দ্বাদশ শ্রেণীর জীবন বিজ্ঞানের যেসব ছাত্র আছো তাদের জন্য আজ আরএনএ কিভাবে উৎপন্ন হয় অর্থাৎ ট্রান্সক্রিপশন প্রসেস সেটা আলোচনা করব। কিন্তু যেহেতু হাতে আমাদের সময় কম, এত ক্লাস দেওয়া এবং আমাকে মনিটরিংও করতে হয় কত ক্লাস হচ্ছে না হচ্ছে, সেজন্য এবং যেহেতু বাংলা মিডিয়ামের ছাত্রদেরও নিট পরীক্ষা অল ইন্ডিয়া বেসিসে হয়, তার জন্য প্রস্তুতি নিতে হবে এবং তার অধিকাংশ ভালো বই ইংরেজিতে লেখা, যদিও প্রশ্নপত্র রিজিওনাল ল্যাঙ্গুয়েজে কয়েকটা হয়, কিন্তু তার যা বাংলা সবসময় সঠিক হয় না, তার সেই বাংলার আবার সঠিক অর্থ দেখার জন্য ইংরেজি ভার্সনটা খুলতে হয়। সেই কারণে আমি বাংলা ভার্সনের ছাত্রদেরও বলবো যে আমি খুব আস্তে আস্তে কথাগুলো বলবো। আমরা রেপ্লিকেশন বা ডিএনএর যে গঠন এইসব আলোচনার সময় যে ইংলিশে আলোচনা করেছি ঠিকই, কিন্তু খুব স্লোলি বলেছি আমি, যাতে ইচ অ্যান্ড এভরি স্টুডেন্ট ইরেসপেক্টিভ অফ দি মিডিয়াম অফ স্টাডিজ হি ফলোজ, আমরা যাতে তাদের কাছে পৌঁছাতে পারি, যাতে সবাই বুঝতে পারে, সেই জন্য সেভাবে বলার চেষ্টা করেছি। যাই হোক, একই পদ্ধতি আজ ফলো করব এবং যেখানটা মনে হবে যে একটু জটিল, একটু বাংলায় বুঝিয়ে দেওয়া দরকার, সেখানে বুঝিয়ে দেওয়া হবে। আমি কিন্তু চাইব যে ম্যাক্সিমাম ছাত্র যারা দ্বাদশ শ্রেণীতে পড়ো তারা এটা দেখার পরে আমি আর একটা গুরুত্বপূর্ণ জিনিস এটা রিলেটেডই তোমাদের দেবো এবং একটা ভিডিও দেবো আমার নিজের তৈরি। আর একটা কথা হচ্ছে যে পড়াশোনা করো, কারণ পরীক্ষা কিন্তু সময়েই হবে। You know that on a DNA molecule, genes are arranged, and every characteristic of an individual depends on the coding that remain in the genes. So DNA produces RNA and RNA produces proteins which proteins are responsible for expression of characters, for phenotypic expressions. Of course, some epigenetic factors are also responsible. তাহলে ডিএনএ তৈরি করে আরএনএ তৈরি করে প্রোটিন, এটাই সেন্ট্রাল ডগমা, আমি আগের দিনে আলোচনা করেছি এবং সেটা আমাদের জানতে হবে যে কী করে ডিএনএ থেকে আরএনএ হয়, পরে আবার আরএনএ থেকে কী করে প্রোটিন হয়, যে পদ্ধতিটাকে আমরা ট্রান্সলেশন বলবো। কেন ট্রান্সলেশন বলবো সেটাও আলোচনা করব। নাও let us begin at a slow pace. The expression of the genetic information contained in a segment of DNA always involves the generation of a molecule of RNA. That means a gene segment always generate a molecule of RNA. DNA replicates itself throughout its length. The whole length of the DNA molecule is replicated. But while synthesizing an RNA molecule, that is called transcription, the whole part of the DNA is not transcribed. Rather, some genes are transcribed. Maybe on a DNA there are many genes responsible for expression of various functions, various characters of an individual, and the transcription of that DNA containing many genes may be simultaneous. More than one gene transcribes simultaneously. At first glance, a strand of RNA may seem quite similar to a strand of DNA, but that there is only difference, that is in the hydroxyl group at the 2' position. In the 2' position, if it is DNA, then there is no hydroxyl group, but if it is RNA, then there is a hydroxyl group in the 2' position and also in the 3' position in both the DNA and the RNA. So it's a difference that at the 2' position of RNA there is hydroxyl group, and another difference that thymine is not there in the RNA. There are only uracils. These are the differences, but these small differences confer on RNA the potential for much greater structural diversity than DNA. A diversity that allows RNA to assume a variety of cellular functions. RNA molecules not only carry and express genetic information, they can also act as catalysts. যখন আমরা সেন্ট্রাল ডগমায় কি বলছি, ডিএনএ থেকে আরএনএ সৃষ্টি হয় এবং আরএনএ কোনো একটা অ্যাক্টিভ পলিপেপটাইড তৈরি করে, ওইরকম এক বা একাধিক অ্যাক্টিভ পলিপেপটাইড মিলে একটা এনজাইম বা প্রোটিন তৈরি হয়, যেটা কোনো একটা বায়োকেমিক্যাল রিয়াকশানকে কন্ট্রোল করে এবং সেইভাবে তার জেনেটিক এক্সপ্রেশন হয়। অর্থাৎ যে জিনটা ডিএনএতে আছে সেই জিনটা তার একটা কমপ্লিমেন্টারি আরএনএ স্ট্র্যান্ড তৈরি করে দেয়, সেই আরএনএটা নিউক্লিয়াসের বাইরে বেরিয়ে এসে, মেনলি মেসেঞ্জার আরএনএর ক্ষেত্রে বলছি, সেই এমআরএনএটা নিউক্লিয়াসের বাইরে বেরিয়ে এসে সাইটোসলের মধ্যে তার ফাংশন চালায়। কীভাবে না, প্রোটিন সিনথেসিস করে। তাহলে কোড, ডিএনএ কোড অনুযায়ী প্রোটিন সিনথেসিস হয়, সেটা আমরা জেনেটিক কোড কীভাবে কাজে লাগে এই প্রোটিন সিনথেসিসে, সেটা আমরা পরে আলোচনায় আসছি। তাহলে ডিএনএ থেকে আরএনএ, আরএনএ থেকে প্রোটিন। প্রোটিন মানে এনজাইমগুলোও তো প্রোটিন দিয়ে তৈরি। তাহলে কখন কোন জিনটার ট্রান্সক্রিপশান হবে, সবসময় সব জিন যে ট্রান্সক্রাইব হতে থাকবে এটা নয়। ধরো তুমি খাবার খেলে, একটা মিল খেলে, তার মধ্যে অনেকটা প্রোটিন খেয়েছো, যত অনেকটা অল্পটা যাই, তাহলে সেই প্রোটিনটা হজম করার জন্য তোমার এনজাইম লাগবে। এটা পেপসিনোজেন হিসাবে ইনঅ্যাক্টিভ ফর্ম, তার অ্যাক্টিভ ফর্ম হলো পেপসিন, যেটা ACL এর সঙ্গে রিঅ্যাক্ট করে অ্যাক্টিভেটেড হয়, পেপসিনে পরিণত হয়। তাহলে পেপসিনোজেন হিসেবে সে তৈরি হবে। কার দ্বারা তৈরি হবে? ওই প্রোটিনটা কার দ্বারা তৈরি হলো? আরএনএর দ্বারা। ওই আরএনএটা কার দ্বারা তৈরি হয়েছে? ডিএনএর দ্বারা। এবং কখন তৈরি হয়েছে? যখন তুমি প্রোটিনটা খেয়েছো তখন হজমের কাজটা শেষ হয়ে যাবে, ওই ট্রান্সক্রিপশন অফ RNA responsible for creating the pepsinogen will stop. এবারে ওইরকমভাবেই শরীরের বিভিন্ন জায়গায় যখন যে কাজটা হচ্ছে, সেই কাজটা লাগানোর জন্যে যে এনজাইমটা দরকার হয়, সেই এনজাইমটা তৈরি করতে হবে। সেই এনজাইমটা তৈরি করার জন্য কে তৈরি করে এনজাইম? আরএনএ। তাহলে সেই আরএনএটাকে ট্রান্সক্রাইব করবে ডিএনএ, সংশ্লিষ্ট জিনটা সেটাকে ট্রান্সক্রাইব করবে। তার মানে একটা জিনিস আমরা বোঝাতে পারলাম যে ডিএনএর মধ্যে অসংখ্য জিন থাকলেও কোনো একটা সেলের মধ্যে কোনো একটা জিন তখনই ফাংশন করবে যখন তার আরএনএর মাধ্যমে প্রোটিন তৈরির প্রয়োজন হয়, আদারওয়াইজ সে চুপচাপ থাকে। অন্য জিনগুলো তখন যেগুলো দরকার সেগুলো কাজ করবে। দিস ইজ দি প্রসেস। RNA is the only macromolecule known to have both informational and catalytic functions. আমরা দেখলাম তাহলে যে ডিএনএর উপর যে কোড আছে সেই কোডটার কমপ্লিমেন্টারি কোড বিশিষ্ট কমপ্লিমেন্টারি একটা আরএনএ স্ট্র্যান্ড তৈরি হয় সেই জিনটার উপরে, সেটা প্রসেসড হয়ে নিউক্লিয়াসের বাইরে বেরিয়ে এসে এটা প্রোটিনকে তৈরি করে। তাহলে ডিএনএ ইনফরমেশনটা প্রোটিনে চলে গেল, ট্রান্সফার অফ ইনফরমেশন ফ্রম ডিএনএ টু প্রোটিন ভায়া আরএনএ। এটা আরএনএর একটা ফাংশন। আরেকটা কি ফাংশন, যখন এই জীবজগতের সৃষ্টির প্রথম পর্যায়ে ছিল, যখন এনজাইম তৈরি হয়নি, তখন জীব সৃষ্টি ঘটছিল কিভাবে ধীরে ধীরে, সেটা হচ্ছে তার ক্যাটালাইটিক ফাংশন। অর্থাৎ আরএনএ খুব খুব কম কারণে হলেও তার একটা এনজাইমেটিক অ্যাক্টিভিটি আছে। সেটা আমরা দেখব যে আরএনএ স্প্লাইসিংয়ের সময় সেটা আমরা বুঝতে পারব। তাহলে আরএনএর দুটো ফাংশন আমরা দেখতে পেলাম, একটা ইনফরমেশনাল, অর্থাৎ ইট ক্যারিজ দি ইনফরমেশন ফ্রম দি ডিএনএ, অ্যান্ড দি আদার ইজ catalytic functions. That means it acts as an enzyme. The discovery of catalytic RNA has changed the very definition of the word enzyme. Many RNAs are complexed with proteins, forming complicated biochemical machines with a wide variety of functions. রাইবোসোমের কথা ধরো, রাইবোসোমের যতগুলো প্রোটিন পার্ট আছে তাদের মাঝে মাঝে রাইবোসোমাল আরএনএ আছে, দিয়ে সেটাকে একটা রাইবোসোম বটিকা, একটা গুটির মতন করে দুটো পার্ট আছে, যার বড় দুটো পার্ট আছে, তোমরা জেনেছো, সেইভাবে ধরে রেখেছে। তাহলে রাইবোসোমাল আরএনএর ওটা একটা ফাংশন। With the exception of the RNA genomes of certain viruses have only RNA. This time we are talking about novel coronavirus, which is also a RNA virus. So in that virus, RNA is synthesized from RNA. That means RNA replicates itself. But sometimes RNA, the viral RNA enters into the host cell and compel the host DNA to form its complementary strand of DNA that again forms the viral RNA. Thus the viral RNA is multiplied inside a host cell. Sometimes this type of viruses have been found, and these are called RNA retrovirus. That means for production of RNA they are dependent on DNA. But RNAs produce RNAs. This is that means they are self-replicating also. But in the process of transcription, an enzyme system converts the genetic information of a segment of DNA into an RNA strand with a base sequence complementary to one of the DNA strands. Not the two strands are simultaneously transcribed, only one strand. And its complementary strand of RNA is formed. Three major kinds of RNA are produced. One is messenger RNA, you know. The other is transfer RNA, tRNA. And the third one is rRNA or ribosomal RNA. And there are also other types of RNAs. I have talked about them in the previous lectures. Replication and transcription. DNA replication. DNA replicates itself. DNA forms its own complementary DNA strand. Replication and transcription means DNA to RNA formation. These two processes differ in one important respect. During replication, the entire chromosome is copied to yield daughter DNAs identical to the parent DNA, whereas transcription is selective. Only particular genes or groups of genes are transcribed at one time. Transcription of DNA can therefore be regulated so that only genetic information needed by the cell at a particular time, at a particular moment, is transcribed. Specific regulatory sequences indicate the beginning and end of the segments of DNA to be transcribed. I am going to the discussion just later. So, the synthesis of RNA is DNA dependent, and the enzyme responsible for the synthesis of RNA is RNA polymerase, but as it is a DNA dependent synthesis of RNA, so this enzyme is actually called DNA directed RNA polymerase. The purified enzyme contains zinc ions as well as magnesium ions. Otherwise, the fundamental chemistry of RNA synthesis has much in common with DNA synthesis. RNA polymerase elongates an RNA strand by adding ribonucleotide units to the 3' hydroxyl end of the RNA chain, as in the case of DNA replication, and thus builds RNA chains in the 5' 3' direction. It is also similar to DNA replication. The 3' hydroxyl group acts as nucleophile. That means it attacks the alpha phosphate of the incoming ribonucleoside triphosphate, and two phosphates, means pyrophosphates, are eliminated, are released, as in the case of DNA. So, a nucleophilic attack is held at the 3' hydroxyl position of the pre-existing ribonucleotide, and the two phosphates of the incoming ribonucleotide, together called pyrophosphate, are released in this process. One enzyme called pyrophosphatase breaks down that pyrophosphate, PP, two inorganic phosphates are there. Pyrophosphatase enzyme breaks down it into two inorganic phosphates, and thus some amount of energy is released, which is responsible for the formation of the phosphodiester bond at the 3' end of the pre-existing ribonucleotide and the incoming, and the 5' end of the incoming ribonucleotide phosphate. In E. coli, a single DNA-directed RNA polymerase enzyme synthesizes all types of RNA. It is a complex enzyme containing 5 core subunits and a 6th subunit called sigma that binds transiently, means for a brief time, to the core enzyme and directs the enzyme to specific initiation sites on the DNA, that is called promoter. These 6 subunits constitute the RNA polymerase holoenzyme. RNA polymerases, whether from E. coli or other organisms, lack a proofreading 3' 5' exonuclease activity as that found in many DNA polymerases. As a result, during transcription, about 1 error is made for every 10 to the power 4 to 10 to the power 5 ribonucleotides incorporated into RNA. Given that many copies of an RNA are generally produced from a single gene, and that all the RNAs are eventually degraded and replaced, a rare mistake in an RNA molecule is of little consequence to the cell than a mistake in the permanent information stored in DNA that we call mutation. Mutation is DNA is more dangerous, but any mutation in RNA, any error in RNA formation matters very little, because RNAs are destroyed after their action, or if not they are acting, they are destroyed in the cell very soon. However, this is one characteristics of RNA. Other differences are also there. Differences between DNA replication and transcription. Unlike DNA polymerase, you have heard of 3 types of polymerases, DNA polymerases. RNA polymerase, I told you that in E. coli there is a single RNA polymerase. So the total name of which is DNA directed RNA polymerase. RNA polymerase does not require a primer to initiate synthesis. Initiation of RNA synthesis occurs only at specific sequences called promoters. I'll be talking about this. RNA synthesis usually starts with a GTP or ATP residue, guanosine triphosphate or adenosine triphosphate, whose 5' triphosphate group is not cleaved to release pyrophosphate, but remains intact throughout transcription. That means RNA synthesis starts with a GTP, guanine, or ATP means adenine. In brief, you call it adenine. These are all triphosphates. All nucleotides are triphosphates. Hence, we call it GTP, guanosine triphosphate, or ATP, adenosine triphosphate. These triphosphates remain there. Triphosphate group is not cleaved to release pyrophosphate, but remains intact throughout transcription. During transcription, the new RNA strand base pairs temporarily with the DNA template to form a short length of hybrid RNA-DNA double helix. This is essential to the correct readout of the DNA strand. Shortly after its formation, the RNA in this hybrid duplex peels off, ছেড়ে সরে যায় ওখান থেকে। পিল করা মানে খোসা ছাড়ানো, যে হাইব্রিডটা তৈরি করছে ডিএনএ আরএনএ হাইব্রিড, সেখান থেকে আরএনএটা সরে খুলে চলে আসে। To enable RNA polymerase to synthesize an RNA strand complementary to one of the DNA strands, the DNA duplex must unwind, as in the case of replication. It must unwind over a short distance because it is the distance of a gene that is to be unwound, and therefore a transcription bubble is formed. During transcription, the E. coli RNA polymerase generally keeps about 17 base pairs unwound, unwinding the DNA ahead and rewinding it behind. Because the DNA is a helix, this process requires considerable rotation of the nucleic acid molecules. Rotation is restricted in most DNAs by DNA binding proteins and other structural barriers, and a moving RNA polymerase generates waves of positive supercoils ahead of and negative supercoils behind the point at which transcription is occurring. This transcription driven supercoiling of DNA has been observed both in vitro and in bacteria in vivo. In the cell, the topological problems which I discussed in case of DNA replication, they are also torsional strain developed, topological problems developed. Here also it is developed. So these topological problems caused by transcription are relieved through the action of topoisomerases. I mean, in case of DNA replication, topoisomerases are required to relieve the DNA molecule of the torsional strain caused due to unwinding of the double helix. Here also, in case of RNA formation, topoisomerases are required. Once the transcription begins, it proceeds at a rate of about 50 nucleotides per second. That means the RNA polymerase, shortly I will call it RNA polymerase, it can add 50 nucleotides per second. Now, the sequences of two complementary DNA strands are different. Complementary sequences are there, and the two strands serve different functions in transcription. A variety of designations are used to distinguish the two strands. The strand that serves as template for RNA synthesis is called the template strand or minor strand. The strand on which RNA is synthesized is called minor strand or template strand. In any chromosome, different genes may use different strands as template. Suppose in a DNA molecule there are two strands, A and B. It is not that all the genes will be transcribed on the strand A or on the strand B. Some genes will be transcribed on strand A and others will be transcribed on strand B. In any chromosome, different genes may use different strands as template. The DNA strand complementary to the template is called the non-template strand. That's the strand on which the RNA is synthesized is called the template strand, and the other strand which does not take part in the transcription of that particular gene is called non-template strand or plus strand. But the plus strand is identical in base sequence with the RNA transcribed from the gene with U in place of T. The difference is only there. In place of T, there is U. So, the non-template strand is also sometimes called the coding strand. Very amazing that the strand that does not function in transcription is called the coding strand. Even though it has no direct function in either transcription or protein synthesis. But this is the convention. So the strand on which the RNA is synthesized is minor strand or template strand, and the other strand that remains ideal for that gene is called coding strand or plus strand or non-template strand. It has 3 names, and the 3 names are conventional. Bacterial promoters are located in the region of a DNA strand just preceding the initiation site of RNA synthesis. The figure is given for you. The nucleotide at which transcription is initiated is denoted as +1. Mind it, the starting nucleotide is called +1, the starting point, and the preceding nucleotide as -1. Those portions of the DNA preceding the initiation site, that is towards the 3' end of the template, are said to be upstream from that site. Those portions of the DNA succeeding it, that is towards the 5' end of the template, are said to be downstream from that site. Analysis of the DNA sequences just upstream from a large number of bacterial genes reveals that 2 short stretches of DNA are similar from one gene to another. One of these stretches is centered at approximately 35 bases upstream from the initiation site and typically occurs as the sequence TTGACA. You look at the figure. This TTGACA sequence is called consensus sequence. That means it is the most common version of a conserved sequence. If it was conserved, that would mean that it is fixed, it is unchangeable, but that is not the case. Some changes may occur in the sequence, but more or less the sequence is the same, hence it is called consensus sequence. Some variations may occur from gene to gene. This site in the promoter, I told you that RNA synthesis starts at the promoter region of the DNA strand. This site of the promoter, this is also known as the -35 region, is recognized by the sigma factor associated with the polymerase. Bacterial cells possess a variety of different sigma factors, which is beyond the purview of our syllabus, however, there are different versions of the -35 sequence, and accordingly there are different versions of the sigma factors. The sigma 70 is known as the housekeeping sigma factor because it initiates transcription of most genes. Alternative sigma factors initiate transcription of a small number of specific genes that participate in a common response. I will give you an amazing example. When E. coli cells are subjected to a sudden rise in temperature, a new sigma factor is synthesized that recognizes a different promoter sequence and leads to the coordinated transcription of a battery of heat shock genes which can produce the required proteins that can enable the organism to tolerate heat. The products of these genes protect the proteins of the cell from thermal damage. This is for your knowledge only. This is not included in your syllabus. However, the second, I told you that there is a consensus sequence TTGACA at approximately 35 bases upstream from the initiation site, and another conserved sequence is also there at approximately 10 bases upstream from the initiation site, and it is called a consensus sequence because it is TATAAT. This site in the promoter, named the Pribnow box after its discoverer. Pribnow discovered this promoter site, hence it is called Pribnow box. You must recall it whenever necessary. This Pribnow box is TATAAT sequence. It is responsible for identifying the precise nucleotide at which transcription begins. Although most mutations in promoter regions reduce the rate of transcription, certain base substitutions in both the -35 and -10 regions can greatly increase this rate. Thus, promoters are more than simply recognition regions. They can act as important control sites in regulating the rate of gene expression. Just as transcription is initiated at specific points in the chromosome, it also terminates when a specific nucleotide sequence is reached. In some cases, a protein called Rho is required for termination. Rho has enzymatic activity that may peel the 3' end of the RNA transcript away from the DNA to which it is bound. In most cases, the polymerase stops transcription when it reaches a terminator sequence and releases the completed RNA chain without requiring additional factors. In E. coli, there are at least two classes of termination signals or terminators. One class relies on a protein factor. I told you that it is called Rho. That means here the termination is Rho dependent and the other is Rho independent. The Rho independent class has two distinguishing features. The first is a region that is transcribed into self-complementary sequences, permitting the formation of a hairpin structure. I am giving you a diagram going through which you can understand it. And the hairpin structure is centered around 15 to 20 nucleotides before the end of the RNA. The second feature is a run of adenylets, hence adenines, in the template strand. I told you also the repeated sequence of adenines that are transcribed into uridylets, complementary strand of RNA. In the complementary strand of RNA, uridines are assembled or polymerized. It is thought that formation of the hairpin disrupts part of the RNA-DNA hybrid in the transcription complex. The remaining hybrid complex, oligo-U, oligo-A, oligo-ribo-U means 15 to 20 nucleotides of uracil, and oligodeoxy-A means adenines of 15 to 20 nucleotides. It is a particularly unstable combination of bases, and this is why the entire complex simply dissociates. And the Rho protein has an ATP dependent RNA DNA helicase activity. Helicase means that breaks the helix, that dissociates the DNA RNA hybrid, and probably disrupts the RNA DNA hybrid formed during transcription. But the detailed mechanism is not known. However, some amazing facts I would like to mention here also. You know that antibiotics are used to kill certain bacteria. Such an antibiotic is actinomycin D. And this, in effect, jams the replication process by inhibiting RNA elongation in intact cells. And there is also another chemical, that is acridine, that inhibits RNA synthesis in a similar fashion. In case of tuberculosis, we use rifampicin. It is also an antibiotic inhibitor of RNA synthesis that binds specifically. In tuberculosis, we use rifampicin as an antibiotic. It binds specifically to the beta subunit of bacterial RNA polymerase and prevents the initiation of transcription. I mentioned these to the use of three antibiotics because sometimes questions arise in your mind as to how antibiotics act, and this is the process how RNA synthesis is inhibited by the use of antibiotics. There are other processes also. Sometimes the cell wall formation around the plasma membrane of the bacterial cells are inhibited by using certain chemicals. These are also called antibiotics or disinfectants.